In 1933, a man whose name we will likely never know was waist-deep in riverbed sediment on the banks of the Songhua River, hauling rubble for a bridge the Japanese military needed built. Harbin, northeastern China. Manchuria under occupation. The bridge was for the Manchukuo National Railway, and the labor force building it did not have the luxury of asking questions. But this man found something in the muck that stopped him cold. A skull. Not a small one, not a fragment. A face, staring back at him from the sediment, almost entirely intact, with brow ridges thick as a man's fist and eye sockets wide enough to fit grown thumbs inside. He knew enough to recognize it was old. The Peking Man discovery had been in the newspapers just 4 years earlier, in 1929, and the idea of ancient human fossils carried a kind of electric public awareness even among laborers working under foreign occupation. He probably knew enough to recognize it was valuable. And that was the problem, because the soldiers overseeing this bridge were not his people, and anything of value found on occupied land belonged to the occupier. So the laborer did something that would shape the next 90 years of human evolutionary science, though he could not possibly have known it at the time. He wrapped the skull, carried it home, and lowered it to the bottom of his family's well. Then, he said nothing. For decades. That skull would sit in darkness for over 80 years, through the rest of the Japanese occupation, through the Soviet invasion of Manchuria in 1945, through the Chinese Civil War, through the founding of the People's Republic, through the Cultural Revolution. The man who found it survived all of it, and he kept his silence through every chapter. Because admitting where the skull came from meant admitting he had worked on Japanese infrastructure, a collaboration that could have destroyed his life under each successive regime. The political cost of speaking was, for decades, higher than the scientific cost of silence. Only on his deathbed, sometime before 2018, did the man finally tell his children what lay at the bottom of the well. They pulled it out, they cleaned it, and they donated it to Hebei University. What scientists eventually found inside that skull, inside a scrap of ancient plaque no heavier than a grain of sand, did not just identify a new fossil. It answered a question that had haunted human evolution for 15 years. What did the Denisovans actually look like? To understand why that question carried the weight it did, you need to grasp how bizarre the Denisovan situation had become. Here is an entire branch of the human family tree, a population that once ranged across thousands of kilometers of Asia, that interbred with our ancestors on at least 2 separate occasions, that left measurable genetic traces in billions of living people today. And yet for 15 years, the sum total of their physical remains amounted to almost nothing. A pea-sized finger bone from a girl who lived roughly 40,000 years ago. A toe bone. A couple of oversized teeth. A partial jaw dredged from a Tibetan cave at over 3,000 meters elevation. That was it. That was the entire Denisovan body, as far as science could confirm. The finger bone that started everything was found in 2008 in Denisova Cave, a limestone shelter tucked into the Altai Mountains of southern Siberia. Russian archaeologists had been digging there for 30 years, pulling out stone tools and animal bones from layers spanning hundreds of thousands of years. The cave sits at an average annual temperature of 0 degrees Celsius, which is partly why biological material survives there at all. Cold is a preservative. Time is a destroyer. 0 degrees is the narrow margin where one force just barely holds off the other. In 2010, Svante Pääbo and his team at the Max Planck Institute for Evolutionary Anthropology in Leipzig extracted DNA from that finger bone and sequenced a genome that matched nothing on record. Not Homo sapiens, not Neanderthal. A third lineage. A sister group to Neanderthals that had diverged from the common ancestors somewhere between 300,000 and 400,000 years ago, spread into Asia, and apparently thrived there for tens of thousands of years before disappearing. Pa Bo, who would later win the Nobel Prize for his work on ancient genomes, called it one of the most exciting moments of his career. And the evidence that followed was staggering in its implications. Roughly 4 to 6% of the genomes of modern Melanesians, the people indigenous to Papua New Guinea and surrounding islands, comes directly from Denisovans. That is not a trace. That is a significant chunk of a person's genetic identity, inherited from interbreeding events that took place perhaps 50,000 to 60,000 years ago. Even more dramatic is what the Denisovans left behind in Tibet. A variant of a gene called EPS1, a transcription factor that regulates the body's response to low oxygen, is found in 87% of modern Tibetans. It is found in only about 9% of lowland Han Chinese. It allows Tibetans to live and work on a plateau where the air has 40% less oxygen than at sea level, without the dangerous thickening of blood that would kill most of us within weeks. That gene variant does not exist in modern Africans. It does not exist in Europeans. It is almost identical to the Denisovan version. The Tibetans inherited the single most powerful altitude adaptation in the human species from a group of people who, until 2010, we did not even know had existed. Consider how strange that is. In 2012, Pabo's team completed a high-coverage genome sequence using less than 10 milligrams of that same finger bone, achieving the first complete ancient genome at a resolution higher than most modern human genomes sequenced at the time. By 2018, researchers had identified a bone fragment in the cave belonging to a girl, Denisova 11, whose mother was Neanderthal and whose father was Denisovan, a first-generation hybrid, proving that these groups did not just coexist, but actively mated within the same cave system. The Denisovans were among the most genetically characterized ancient populations on Earth. And yet, we could not draw their face. We had 30-fold genome coverage from a bone fragment smaller than a penny. We could model their population splits, map their gene flow across 2 continents, and identify their immune system contributions to living humans. But ask a paleoanthropologist to sketch what a Denisovan looked like, and the honest answer was a shrug. That is what made the Harbin skull so explosive when it surfaced. The skull that came out of that well in 2018 was unlike anything in the existing record, nearly complete, missing only the lower jaw. A brain volume of approximately 1,420 cubic centimeters, squarely within the range of modern humans. Massive brow ridges set above wide, deep eye sockets. A broad nose. A flat face with cheekbones that fell somewhere between the heavy architecture of a Neanderthal and the more delicate proportions of Homo sapiens. The skull was almost a foot long, making it the longest hominin cranium ever measured. The estimated body weight of the individual it belonged to was around 100 kilograms, roughly 220 pounds. This was not some graceful forest dweller. This was a cold-adapted heavyweight. The team that first described the skull in 2021, led by paleoanthropologist Chang Ji at Hebei Geo University, proposed it as a new species. They named it Homo longi, the Dragon River human, after Heilongjiang province. The naming was deliberate and provocative. The skull had a mosaic of features that resisted clean classification. Ancient traits like the low, elongated brain case echoed Homo erectus. Modern traits like the flat face and expanded brain capacity pointed toward something closer to Homo sapiens or Neanderthals. The team argued these combinations justified a separate branch on the evolutionary tree, a lineage that included the Harbin skull and possibly several other hard-to-classify Chinese fossils from the same murky period that paleoanthropologists call the muddle in the middle, the confusing stretch between 1 million and 300,000 years ago, where fossils refuse to sort into neat categories. But from the moment the skull was published, many researchers suspected a different answer. They thought it was a Denisovan. The geography was right. The age was right. The physical proportions were consistent with the Qiaomei jaw from Tibet, the only Denisovan specimen ever identified by protein analysis at that point. The problem was proof. And proof, in this case, meant DNA. The obvious approach was to extract genetic material directly from the bone or from the single tooth still lodged in the upper jaw. Standard ancient DNA work targets the petrous bone, the dense pyramid-shaped structure in the inner ear that is the gold standard for preserving ancient molecules. But there was a complication that perfectly encapsulates why the Harbin skull spent so long in scientific limbo. Chinese collectors had removed the petrous bone sections decades earlier. Nobody recorded when or why. The tooth yielded nothing usable either. Every conventional extraction route was a dead end. A skull that had survived Japanese occupation, civil war, 80 years of well water, and the Cultural Revolution had apparently lost the one piece of itself that modern science most needed. That is when Qiaomei Fu made a decision that shifted the entire trajectory of this discovery. Fu is a paleogeneticist at the Institute of Vertebrate Paleontology and Paleoanthropology in Beijing, part of the Chinese Academy of Sciences. She had been among the first researchers to study Denisovan DNA back in 2010. She understood the Denisovan problem better than most. And she knew that when bone and tooth have failed, there is one last place ancient DNA sometimes hides: dental calculus, the hardened plaque that accumulates along the gum line over a lifetime of eating. It is not a standard target. Bone is denser, teeth are harder, and both typically preserve far more genetic material. But calculus has one crucial advantage. Its crystalline mineral structure entombs whatever biological material it contacts during life, sealing cells, bacteria, and fragments of host DNA inside what Harvard archaeogeneticist Christina Weriner has compared to a molecular time capsule. The crystalline lattice resists the degradation that destroys DNA in other tissues. In specimens this old, Weriner has noted, calculus can be the only hope. Fu's team found a thin ring of blackened calculus around the root of the Harbin skull's single remaining molar. They scraped 0.3 milligrams from it, a third of a milligram. To put that in perspective, a single grain of table salt weighs about 1 milligram. This sample was a fraction of a grain of salt, and it had been sitting in plaque on a dead man's tooth for at least 146,000 years. When the team processed the extract, the results were messy. Nearly 80% of the human genetic material they recovered was modern contamination, DNA from the researchers and collectors who had handled the skull over the decades. Shijun Ni, one of the co-authors on the original Homo longi paper, acknowledged that some of the contamination was likely his own. But contamination is not the end of the story. It is a well-understood problem in ancient DNA research, and the protocols for filtering it out are robust. What mattered was the 20% that was genuinely ancient. This is where we should talk about what this channel exists to do, which is to track exactly these moments where one piece of evidence reconfigures an entire field. If that matters to you, subscribing means you will not miss what comes next, because the Denisovan story is developing across half a continent right now, and we are following every thread. From that filtered fraction of ancient material, Fu's team reconstructed the mitochondrial genome, the smaller, maternally inherited DNA sequence that serves as a lineage marker. They found 27 gene variants in the Harbin specimen that exist only in the 7 known Denisovan individuals previously sequenced from Siberian sites. 27 specific molecular signatures. None of them arise from modern human contamination. None of them are shared with Neanderthals. The mitochondrial DNA placed the Harbin individual on an early Denisovan branch, genetically closer to the older Denisovan specimens from Denisova Cave than to the more recent ones. That pattern is consistent with the skull's estimated age and with the 3,100-kilometer distance separating Harbin from the Altai Mountains. Migration takes time. Genetic drift accumulates over distance. The molecular clock and the map agreed. Simultaneously, a parallel study published in Science attacked the question from the protein side. The team analyzed samples from the Petrus bone region and recovered 95 endogenous ancient proteins, including more than 20,000 peptides and 122 single amino acid polymorphisms specific to the Homo genus. Within that dataset, they identified 3 protein variants unique to Denisovans, matching specifically with the Denisova 3 specimen from the original cave. 2 completely independent molecular lines of evidence from 2 different biological substrates processed with different techniques in different laboratories, converging on the same conclusion. The Harbin skull is Denisovan, and that identification does 3 things simultaneously. First, what the evidence shows. This is the first nearly complete hominin cranium ever molecularly confirmed as Denisovan. Its dimensions are extraordinary. The skull stretches almost 12 inches in length. The brain cavity holds roughly 1,420 cubic centimeters of volume, placing it comfortably within the modern human range and above most Neanderthal averages. The brow ridges are among the thickest on record for any late Pleistocene hominin. The face is wide and flat, combining features that researchers had previously distributed across 3 or 4 separate species. The individual likely weighed around 100 kilograms. We now have, for the first time, a Denisovan face to set beside the Neanderthal faces we have studied for over a century. Second, what it rules out. Before Harbin, it was technically defensible to argue that the Denisovans were a genetic abstraction, a population label applied to scattered fragments that might not represent a coherent physical type. The genome from the finger bone could not tell you if Denisovans in Siberia looked anything like Denisovans near the South China Sea. The fragments were too few and too broken to build a morphological profile. The Harbin confirmation eliminates that ambiguity. The skull's physical features are consistent with the Qiaomei jaw from Tibet and the Penghu jaw from the Taiwan Strait, both of which have been flagged as likely Denisovan based on protein or morphological comparisons. We are now looking at a population with recognizable shared traits distributed across an immense geographic range. That is not a statistical artifact. That is a species. Third, what it implies. A 100-kilogram hominin living in northeastern Asia during glacial periods faces caloric demands that researchers estimate at over 4,000 calories per day. Meeting that demand in a periglacial environment requires organized hunting, probably of large herd mammals like deer and horse. It implies seasonal migration patterns tracking prey across hundreds of kilometers. It implies group coordination, food sharing, and the cognitive infrastructure to sustain all of it across generations. Isotopic analysis of the Qiaomei jaw has already hinted at a meat-heavy diet for Denisovan populations on the Tibetan Plateau. The Harbin skull adds body mass to that picture, and body mass has consequences. Large-bodied hunters in cold environments do not survive by accident. The debate, predictably, has not settled. The taxonomic question is real, and it is not merely academic. Chris Stringer at the Natural History Museum in London, who has collaborated on analysis of multiple Chinese hominin fossils, accepts the Denisovan identification but argues that Homo longi should remain the formal species name for the group. In his framing, Denisovans are Homo longi, the same way New Yorkers and Beijingers are both Homo sapiens. The species name came first, and taxonomic priority matters. Svante Pääbo, the geneticist whose lab defined the Denisovans in the first place, resists separate species names entirely for late Pleistocene hominins, pointing to the extensive interbreeding between groups as evidence that clean species boundaries did not exist. Bense Viola, a paleoanthropologist at the University of Toronto who has conducted excavations in Denisova Cave itself, takes a more pragmatic view. He finds the combined protein and DNA evidence compelling and has noted that the identification will make research far easier going forward because scientists can now compare unknown specimens to a confirmed skull rather than to a collection of fragments and isolated teeth. But this identification does more than resolve a dispute about one fossil. It opens a cascade of potential reclassifications. The Dali skull from Shanxi province, found in 1978. The Jinyushan cranium from Liaoning. The Hualongdong specimens from Anhui. These are Chinese fossils from the Middle Pleistocene that have bounced between classifications for decades, sometimes labeled Homo heidelbergensis by convention, sometimes left in outright taxonomic limbo. The Harbin research team's own analysis suggests that many of these fossils share morphological features with the Harbin cranium. If Harbin is Denisovan, then the probability increases substantially that some or all of these specimens belong to the same lineage. What was once the muddle in the middle, an incoherent jumble of unclassifiable fossils strewn across Chinese museum collections, may turn out to be the Denisovan fossil record that was there all along, waiting for a molecular key to make sense of it. And a smaller but vocal faction questions the skull's provenance altogether. Because the laborer removed it from its geological context in 1933, we have no stratigraphic information, no associated tools, no faunal layer to cross-reference. The uranium-thorium dates on the skull itself span a range from 62,000 to nearly 300,000 years, and the statistical best estimate of 146,000 years carries real uncertainty. Without context, some scholars argue the evolutionary placement is built on sand, however molecularly sound the identification might be. The molecular evidence, though, is the part that does not bend. The DNA is Denisovan, the proteins are Denisovan, and the technique that unlocked them carries implications beyond any single skull. Dental calculus, the material that dentists spend their careers removing, turns out to be one of the most durable archives of ancient molecular information in the fossil record. Museums around the world hold thousands of unclassified fossil teeth from the Middle Pleistocene, the exact period where the muddle in the middle has frustrated researchers for decades. Many of those teeth carry visible calculus deposits. Most of those deposits have never been sampled. The Harbin skull spent 80 years hidden in a well. The method that identified it may have been hiding in plain sight even longer, clinging to the roots of teeth in collections from Beijing to Berlin to Nairobi. There is a version of the EPAS1 gene inside the lungs of a Tibetan Sherpa climbing above 8,000 meters right now. That gene was passed down from a Denisovan ancestor who lived somewhere in Asia tens of thousands of years ago. It allows the Sherpa's blood to carry oxygen without thickening into a dangerous sludge that would trigger stroke at altitude. In Melanesia, children carry immune system variants traceable to the same Denisovan lineage, inherited from encounters between populations that happened before the last ice age ended. We carry pieces of these people in the way we breathe, the way we fight infection, the way some of us tolerate conditions that would incapacitate others. The Harbin skull sat in darkness because a man in an occupied country decided to protect something he could not name through decades of upheaval that would have erased it entirely if he had spoken. The Denisovans themselves have lived in a different kind of darkness for 15 years, known only by their code, never by their faces. Both silences are ending now. One ended with a deathbed confession. The other ended because a geneticist in Beijing scraped 1/3 of a milligram of tartar from a dead man's tooth and found a species inside. If museums hold thousands of unsampled fossil teeth with visible calculus on their roots, how many Denisovans have been sitting in storage drawers for decades, already found but never identified?